So a few caveats. So some of what I'm going to describe is, I'm going to say it's only marginally HPC, in the sense that the computations could be done on a single node. A lot of the work would be embarrassingly or perfectly parallel, but the computation was still a very, very important component of the project. And compared to some of the other things I've worked on, this work is, I consider it to be very easy. It didn't require anything really cutting edge, just some very basic cache level optimization, standard performance tuning techniques. But that said, the collaboration, I think what was really valuable about this is that it shows how big an impact we can have on researchers in fields that are relatively new to high performance computing. So again, the engineers, the physicists, the molecular biologists, they've been doing HPC for decades. A lot of them have picked up the techniques that they need in order to develop the efficient parallel code. But in a lot of these newer fields, the life sciences, the social sciences, humanities, ecology, finance, there's often a lot of relatively easy work that can be done that can have a really big impact. So what drives a lot of this work is that we want to understand the environmental impact of new projects. So in the United States, and I'm sure it's the same thing in Australia, every time we have a new project, a building, a road, a power plant, malls, pipelines, dams, airports, harbors, they have to undergo an environmental impact study. But we find, though, that sometimes a project, it may pass the study. It could get the green light to go forward. There are sometimes faulty predictions. And those could have very, very big impacts on both the environment and on a company's bottom line. So in our case, we're looking at possible placement of a wind farm. And we want to make sure that we get this right, because we don't want to end up killing wildlife, in particular these California condors, and we don't want to have to move a wind farm after it's been installed. That would be a very costly process. And for this particular work, we're going to be extending into 3 dimensions. This is something that's not normally done with the wildlife tracking. And I'll show how this can reduce the likelihood of having false positives or false negatives, in this case, particularly false positives. So a little bit of background. Much of this work was made possible by what my colleagues call the biotelemetry revolution. And namely, this is the miniaturization of these biotelemetry devices. So if you look at the 2 pictures here, we have a California desert bighorn sheep, and next to it is some kind of a bird of prey that's a hawk or a falcon. And if you look at the bighorn sheep, you'll see the very large transmitter that's around his neck. I've never held one of these, but I'm going to guess that this probably weighs in the order of pounds. It has a very, very large transmitter in there. There's a little antenna, a large battery pack. And you could do this for a large, strong animal like a bighorn sheep. These weigh several hundred pounds. They're extremely strong. They can climb very, very rough terrain. But of course, you're not going to be able to take something like that and put it on a small animal, a small mammal, a bird or a lizard. And these new devices that we have are incredibly small compared to the older technology. So it shows here on the left what one of those devices next to a quarter. And I think the American quarter is about the same size as the Australian quarter. This is very small, maybe half an inch across. And I believe that the smallest devices only weigh about 5 grams. So with this miniaturization, there's a lot wider range of animals that we can put these devices on. And there are a lot of companies that are making these devices. So this is just a sampling that my colleagues at the zoo and the U.S. Geological Survey gave me. You can see from the logos that a lot of these are still aimed at larger species. There's one with the polar bear in the upper right, and what looks like a reindeer or caribou in the up center. But you'll notice that a lot of these, they're also specifically having smaller animals. There's birds, there's fish, small mammals, reptiles. So what we want to do with the data that we're collecting from these devices is that we want to turn discrete observations into home ranges. So unlike your GPS device or your iPhone, which is giving you a continuous tracking, but with these devices we get discrete readings, and they're typically spaced about an hour apart. And even then, we'll typically have missing data. Sometimes we might miss a reading. We'll go, say, 2 hours or 3 hours between readings. Now, ideally, we would love to have continuous data, but it's just not possible because, as you know, once you turn on the navigation feature on your smartphone, it draws a lot of power. It drains your battery very quickly. For us, we can always keep our phones plugged in, or we can bring them home and recharge them. But of course, with animals that we're releasing into the wild, we don't have that luxury. We either have to use very small batteries, or we can partially recharge them, say using a small solar cell that goes along with the device. But that gives us very limited power. So getting readings, say spaced an hour or so apart, is about the best that we can do. So what we want to do is take these discrete readings, X, Y, Z coordinates, and we want to turn them into home ranges, essentially estimates or probability distribution functions of where the animals are spending their time. So I'm going to start with an example in 2D, and this is the way most of the work is done. This was something that one of my colleagues put together. I think he just walked around town, and he would periodically mark his location. So those are all the little red flags that you're seeing. And then he applied one of the standard 2D algorithms and turn this into a home range estimation. So as you would imagine, the darker colors, the dark red is where he's most likely to be spending his time, followed by the orange, the yellow, and then eventually the green. So note though that these are probabilities. We really don't know what his excursions were between those points. We know exactly where he was, or to within the error of the GPS device, where he was at those discrete measurements, but we're trying to estimate where he was in between and create this home range estimate. So what was really unique about this work, and it was essential if we really wanted to understand the lifestyle, the motion of the California condors, was go from 2D to 3D, and this work, extends it from a 2D, which is fine for some animals, say for large terrestrial animals. But once we start trying to track animals that swim, fly, or climb, this is going to be inaccurate. So I know it's a Friday morning. Nobody wants to see a lot of math, so I won't spend too much time on this. If you really want to know the formalism, you could take a look at the paper that I referenced at the bottom, the Tracy et al. That was published in PLOS One last year. And then we also described this work at the annual Exceed conference last July. The first paper focuses on the math. The second one focuses on the computation and the optimization of the software. So just to go through this very quickly, we start off, we come up with a kernel function that describes the probability density at a particular point at a given time. So you could think of this as being a Gaussian, a multidimensional Gaussian centered at a given point, which then trails off as we get further away from the origin. And then we're going to calculate an average probability density in each voxel over a time interval, say from Tm to Tm plus 1. And then finally, we're going to sum that over all time steps to get the total probability density for that voxel. And the variances, the sigmas that you see in the first equation, they depend on the time difference between the observations and the time at which the kernel was calculated. So we calculate these kernels not just where we have the measurements, but at interpolated time steps. And as you can imagine, as we get further away from one of those discrete GPS readings, we're going to have more variance, more more uncertainty in where the animal is. And then the means, mu1, mu2, mu3, they depend on the difference between the locations of the measurements. So to make this a little more concrete, I'm going to draw this in 2D because I've never figured out a good way to do this in 3D. Imagine that we have 3 points, T1, T2, T3, where we've made our measurements. So we're then going to draw straight lines between those points, and we're going to break that up into steps. So we're going to have T1 plus delta T, T1 plus 2 delta T until we get all the way up to T2. And then we're going to do the same thing with each set of sequential time points. And then if you look at the line between T2 and T3, you'll see that I have some dashed circles around each of those points where we're evaluating the kernel. So you could think of that as representing the uncertainty in the position. So when we're right at point T2 or T3, we're very sure where the animal is. But as we get to the midpoint between those 2 time steps, there's more and more uncertainty. We're doing the interpolation about points drawn on the straight line between the 2, but of course there could be a lot of excursions from that straight line. And then once we do this, at each of those points, we're going to calculate this kernel, and we're just going to make the contributions to each of those grid cells, or pixels in 2D, voxels in 3D. So where we found it important to do an accurate 3D treatment is, again, for these animals that have the large vertical excursions. So of course, birds, mammals like pandas, which live in mountainous regions and do a lot of climbing up and down the hills, and of course aquatic animals. In this case, that is a dugong in the last panel, which I believe lives in South Asia, and it's very, very closely related to an animal we have here in the U.S., the manatee, which is found in Florida. So looking at an example for the manatee first, we were able to use this on the GPS data that we collected from the manatee to collect home ranges for both low tide and high tide. And this is really important. These animals live in coastal areas, and I believe they are endangered. I know the manatee in the United States is. And I don't think anybody really hunts these animals, but they're killed often by collisions with boats. So they could be hit by boats, they could be cut by propellers. So we'd really like to know where they're spending their time. So with this reconstruction in 3D, we're able to get a good feel for where they are, when they're near the surface, where they are at different times of the day, and hopefully route the boats in such a way that they're not going to be colliding with the dugongs. For the case of the panda, we really consider this to be a 2.5D reconstruction. So of course pandas don't fly, they don't swim that I know of, but they live in very rugged mountainous areas. And when we did the reconstruction of the home ranges using this more accurate 3D model, we found a very important result. And that's that if you look at that surface area that they cover, it's actually about 30 to 50% larger than we had expected using a simple 2D model. So of course that's a very important result. We realized that these animals they're known to need a lot of land, but it turns out it's even significantly more than we had expected. What I'm going to turn attention to now is the real focus of this work was with the California condors. So these are native to the Southwest United States and northern Mexico, and they're absolutely fantastic birds. If you ever get a chance to visit the San Diego Zoo or the San Diego Wild Animal Park, they have a few of these on display. They may be one of the largest birds in the world, right up there with the Andean condor, and they were critically endangered. At one time, there were only 22 of these remaining in the wild. That was back in 1987. And they made what was considered at the time a very controversial decision to collect all of the remaining condors and breed them in captivity. So it's a tough decision. There are a lot of trade-offs. Of course, when you're trying to capture animals in the wild, there are dangers with doing that. But on the other hand, they were so critically endangered in the wild, down to just 22 remaining wild condors, that they most likely would have gone extinct if this hadn't been done. They started reintroducing them into the wild in 1991, and now the total population is still small. This is a little bit out of date, but as of last year, they were up to 439, and that's roughly equally distributed between wild and captivity. So one of the reasons that they started going extinct to begin with was poaching. So of course these birds, they're scavengers. They don't kill live animals. But a lot of ranchers thought that the condors were killing their cattle. They go out in the field, they'd see the condors feeding on carcass, and they started shooting the condors. And of course, through education, they learned that condors are not killing their cattle; they're just eating the ones that had died. But more recently, there's of course problems with habitat loss, electrocution from power lines. They're also subject to lead poisoning. In California, we've banned the use of lead shot in the regions where the condors live. A lot of times the hunters will, they'll kill a bird or small game. They might not retrieve the animal. The condors will come by and they'll eat the carcass, and they'll ingest those lead pellets. And one of the new threats is wind farms, where we're trying to develop a lot of wind energy in the U.S. Unfortunately, condors and wind farms like the same places. You always put a wind farm where it's windy, and condors likewise like to sail on these wind currents. So just a little bit more about the captive breeding program. This is one of my colleagues, James Shepard. I think he has one of the most interesting jobs. Most of us type for a living. He actually goes out in the field. He collects these condor eggs. Amazing work. I've seen pictures of him rappelling off of cliffs to get these eggs. So he's part of the team that collects these in the wild, and then they're then brought to the San Diego Zoo and the Los Angeles Zoo as part of the captive breeding program. The birds are then raised in captivity. You can see here on the left a condor chick being fed by using a hand puppet. And the reason we do that is to avoid inappropriate imprinting on humans. Some of you might have seen this picture here on the right of Konrad Lorenz, a very, very famous psychologist or cognitive scientist. And he did some early work on imprinting, but particularly on geese. And he found that if you would take geese when they were just hatched, he could get them to imprint on him or his boots rather than the adult geese. So we, of course, wanted to avoid that with the condors. We want them to have a healthy fear of people. And then eventually, if all goes well, we get to release these back into the wild, and in several locations throughout the Southwest. If you look on the map here, this little green blob, I'm not sure if you're seeing my arrow in the corner of, in that southwestern corner of California, that's San Diego, very, very close to us. Below that is the release site in Mexico, large range up in central California, and a couple other ranges in Utah and the vicinity of the Grand Canyon. So for the condors in particular, we're interested in how they interact with the wind farms. So we built a 3D model of the wind of a wind farm using the industry specs for the turbines. And we placed them in one of the condor release sites. This is a national park in Mexico, Sierra de San Pedro Martir National Park. And if you look on the picture on the right, kind of in the middle, that gray peak, that is Picacho del Diablo. It's, I believe, the highest peak in Baja California. It's about 3,000 meters, very rugged area, and the condors love it there. So let me just skip over to the next slide. So using this model wind farm placed in the condor release site, we wanted to know what would the condors who are living in an area, the ones that have been tagged where we have the GPS data, what would they be colliding with these turbines? So if we did the calculation using the 2D kernel density estimator, of course we would have found that they do collide. when we did this in 3D, we found that the condors safely flew well over the tops of the turbines. And again, you might be wondering, if you look at the condor home ranges shown here in red in panel A, why would you want to put a wind farm in that particular place, right where the condors are? And again, it's because condors and wind farms, but both require wind. We typically put these wind farms in the mountain passes, and it's also where the condors like to fly. Okay, so getting to the computation, I've talked about the 3D, why the 3D is superior to 2D, especially if the animal is making large vertical extensions. But of course, the drawback to 3D is that it's going to be a lot more expensive. I'm sure a lot of you have done 2D and 3D simulations, CFD and molecular dynamics, and so on. And you realize that once we go from 2D to 3D, it's an order n squared problem to an order n cubed problem. It's become much more computationally intensive. In addition, the function that we need to evaluate in 3D is more complicated. I mentioned earlier you could think of it as a 3D Gaussian. It's actually a little more complex. It's a Gaussian in 2 dimensions, but in the 3rd dimension, it's more complex function involving error functions. So we started working with the folks at the zoo. They wanted to know if we could improve the performance of their code. These were all obtained using a single node of the Gordon supercomputer. And for those of you who aren't familiar with Gordon, the standard node is the dual socket Intel Sandy Bridge, each with 8 cores. So we have 16 cores total, each capable of doing AVX instructions. The original version of the code was written in MPI, but I actually rewrote this in OpenMP to make it a lot simpler and take advantage of the fact that we could handle different data sets in parallel. So when we compare when we look at the original version of the code, using their standard benchmark, what they consider a condor bird month, so 337 observations of a single bird over a 1 month period, we found that this took about a half an hour to do. So, not very long. It's not a very big calculation, but keep in mind that this was just 1 month for a single bird. And we'd like to extend this to many birds for many months, and even extend to more common species than condors, say maybe thousands or tens of thousands of migrating geese. So I was able to optimize the software, again, using some very basic techniques. And instead of having a loop over calls to a subroutine, I pushed that loop inside subroutine, did some pre-calculations of quantities. This is very nice. It's a uniform 3D grid. So a lot of the calculations that we do in the vertical direction, we can do once. Say these complex error functions and exponentials. We can save them and we can reuse them. And then also some basic cache level optimization. And by doing that, and this required no changes to the underlying algorithm or it didn't impact the results at all, we're able to get a speed up of about 43x. So we've gone from something that took a half an hour to about a minute. And another thing that we noticed in their code is when they would calculate one of these kernel functions, instead of just looking at the contributions to nearby cells, they would look at the contributions to the entire 3D domain. And if you look at how quickly these die off, you really don't need to extend the calculations out that far. In fact, if we use a very, very big cutoff, d equals 100 means that I'm extending out 100 cells from the center of where the kernel's being calculated. This is so large, it's much larger than you need to do. That gave us another 10x speed up. So we've gone from something that took about a half an hour to something that's taking about 4 seconds. And then we find if we're willing to do tight cutoffs, so I wouldn't necessarily do this if I were going to design a wind farm, but for, say, the biologist in the field, if he identifies a bird, wants to get a quick estimate of the home range, if we use a tighter cutoff, which still gives answers that are very close, to the full resolution answers, we can do the calculation in well under a second. So we've gone from being a supercomputing problem to an iPhone problem. But we didn't want to stop there. But whenever I work with a group and we get a big improvement in performance, we would start talking about what are the problems that you really, really want to do, the things that you thought were just kind of crazy, that wouldn't be doable with the current version of the code. So I like to say that tough problems become easy, insanely hard problems become doable. So we decided to combine the computation and the visualization. I've been focusing just on the calculation of these home ranges. We want to combine it with visualization of the terrain and get a more accurate picture of how the animals are interacting with their environment. So I'm showing a snapshot here. I know it's a little bit hard to see. In panel A, you'll see some thin red lines. Those are straight line trajectories between the points where we have the observations. And then as we go to panels B, C, D, E, F, we're looking at different thresholds for the probability. So if you come from a chemistry, for example, chemistry or physics background, I like to think of these as electron probability distribution functions. Except in this case, it's home ranges for the condor. So the dark purple in panel B is where the condor is spending most of its time. If we increase the probability threshold, we get panel C, and so on and so on. So what we would really like to do, though, is not just single snapshots. We'd like to do a more challenging problem. In particular, we wanted to see if we could make a movie for a pair of breeding condors. And instead of doing just one snapshot, we decided to take a 2-day window. So we calculate the home range using all of the points, all of the readings within that 2-day window, and then slide this in 1-day increments. So this particular problem, for one pair of breeding condors, for the data spanning, I believe, about 2 years, would have taken more than 1,000 core hours on Gordon using the original version of the code. And that's not accounting for all the errors and false starts and parameter explorations that you have to do. And of course, with this kind of a visualization or calculation, there's going to be some, it's going to take some work until we figure out how wide that sliding window should be, what the increment should be. So I'm just about done here. I'm going to show a movie result of for this breeding pair of condors. Once I start the movie, in the lower left, you're going to be seeing the home range for the female. In the lower right, for the male, the upper left is going to be both of the home ranges at the same time. Make sure the movie comes up. I'm just going to pause for a minute. So again, the upper left, you're going to be looking at the home ranges for both birds at the same time. I'm shown in different colors, blue and orange. Lower left is going to be the female. Lower right is going to be the male. Upper right is going to be the trajectories, just straight line trajectories between the readings. And I've looked at this movie probably 100 times now. Every time I see it, I see something new. You'll notice that most of the time the condors are tracking together. But especially toward the end of the movie, you'll see a lot of times when they are going out in different directions, I imagine, looking for food, and then returning to the nesting area.
But Bob, is the nest in the middle of that distribution? Is that?
Yes, it is. And that's it. I'm going to wrap up there. I just want to acknowledge my collaborators on the left. That's James Shepard, who you saw earlier with Condor Egg, and Jeff Tracy. Again, Jeff was developer of the 3D algorithm. The visualization was done by Amit Chirazia, and I think David knows Amit. Amit is a absolutely phenomenal visualization expert. And then a couple of my colleagues at SCSE, Glenn Lockwood and Mahidar Tataneni. And I'm not going to go through the list of all of the organizations that supported this work, but I do want to point out that it was a very broad set of funding agencies, including the U.S. and Mexican government agencies, industries, Sempra Energy, and partial support by the National Science Foundation. And with that, I'm going to wrap up, and I'm happy to take any questions.